this topic for today is about finding your ancestors using DNA. And of course, it's a bit difficult, DNA. It can be very confusing if you're a newcomer to this whole thing. And you really begin to wonder whether DNA tests are useful, are they accurate, these sorts of questions. What's the best test to take? Who should you put your swab into? And what do I expect back when I, put them in? Is one company better than another? These are the sort of questions we're going to look at today. And we have with us Ali Macdonald. I was just going to hope I get, yep, Ali Macdonald. And some of you will have met Ali at the last gathering we had in Inverness. Now, this is a picture of us in Inverness, right? This is Inverness Castle, just to prove where we are, up the back here. Where's Ali? That's what I want to know. Come on, anybody got any guesses? This fella here was the fella who gave the vote of thanks at the last Scotland meeting. You recognise some of the other people? Come on. Someone's got their microphone on. Peter. And, well, this is Linda here, now Ali's wife, at that time, fiance. And here he is hiding behind a good woman.
Yeah.
You know that, Barbara, don't you? And of course, you recognise some of these people. Actually, Star isn't here today, is she? So now you can't get a photo of Ali without him running around somewhere, like in this sort of lovely, lush countryside. You get Ali running around. Of course, I should say that he is such a professional at this area that we're going to talk about today. We're so lucky to have him. He's a teaching fellow and lead tutor at the Department of Genealogical Studies in the Centre of Lifelong Learning at the University of Strathclyde. He's been involved in family history research for over 30 years and has a particular interest in the Highlands. So he's the right person for us to be talking to us today. His research focuses on genealogy through DNA, and he's currently researching the origin and linkage between male lineages in British Isles and the Y chromosome DNA. He is as well the administrator of the Clan MacLennan DNA Project, an administrator of Clan MacLennan DNA Project, and also a number of other projects, including the Scottish DNA Project, which of course is very important to the overall picture. So I got to say to you though, that I first met Ali in 2014 when I met him at the Bannockburn reenactment, the 300-year Bannockburn anniversary. And he sold me a kit. He also sold Rory a kit. So Rory's and my kits are next numbers to each other. But then when I got the results back, I was quite excited. But for some time, like a year or two, I didn't have anybody who was close, anything like my profile from my DNA sample, except Ali. He was really close to me, had the same numbers. Well, I thought there's some mistake here. He got the samples mixed up or something, but no, it turned out there was a bit of a difference after he did some more research. But he and I actually, for a long time, were the closest people that I could, he was the closest person I knew. I later have had a test done with a person I know is my relation who lives in Vancouver, and fortunately we matched up exactly. So that was pretty good. So there we go. That's Ali. Now I'll just hand over to Ali now, and he can take it from here. I'll stop sharing, Ali. If you could be ready to share, that'd be good. There you go. Ali, over to you.
Okay, thanks very much, Bruce, for the introduction. So this afternoon, you can all see that, hopefully. We're going to step our way through the complexities of DNA testing. Now, fortunately, a lot of you submitted questions, and I've sought to fold those into the presentation today and try to answer the questions as best I can. Now, this topic is very large, it's vast in fact, and we're not going to be able to cover everything today. In fact, you're going to get far too much information and you're going to have to just be patient and see this as an introduction. Some of you I know have done testing already, but you have lots of questions, and we'll hopefully find our way through it today. There'll be opportunities for some questions during the time, and certainly at the end, when anybody wants to hang around, there'll be opportunity to ask more questions. So Bruce has thankfully introduced myself. I can't really say much else. I first got tested back in the early 2000s, and I got addicted to it. And I got addicted to it because it produces, DNA testing produces new evidence that we don't have previously. We didn't have previously. And I think that's one of the things that I'm so enthused about. But we have to use this technology, this tool correctly. And I think that's an area that needs education, and even knowing what is the right test to take, what is the difference between the tests, can take a little bit of time to get your head around. So, without too much ado, Bruce mentioned Bannockburn and how we met there, and at Strathclyde we have a website where we have the findings of the Bannockburn, or some of the findings of the Bannockburn research, and we're currently working on research which is similar to do with the Declaration of Arbroath, which obviously the 700th anniversary was this year of the letter that was sent to the Pope, where the nobles, the important people in Scotland at that time, sought to have the Pope's blessing on Scotland becoming continuing to be an independent country. So I'm able to show you just a couple of links to that, just in case you want to go back to it later. I won't be jumping back and forward too much. This is the Declaration of Arbroath project. If you just Google Declaration of Arbroath, University of Strathclyde, this page would come up, and explains all the current research that we're doing, who's involved, what lineages we're looking at, the different families. And if we were to look at the other one, if I can find it, oh, here we are now. This is the Bannockburn research. Now, there's much more material here for those who are interested, and I'll just go back one there. That's better. You can see that now. So what we have is the Bannockburn research. Some of the individuals we researched. If you click on them, so I'll just go to Angus Hogg MacDonald. There's a biography, and there's a coat of arms there. And if you click on the family tree, the genealogy comes up, and some of that you might find quite interesting. So the current research on Arbroath is again looking at Y chromosome research. It's researching the individuals who had their seal or who were signatories to the letter, and we are, as well as researching the male line descents that are alive today or are still extant today, and then take a DNA sample and we link those individuals up with folk who don't know their ancestry, who have only got to the 1780s and there's no paper trail. They carry a name of one of the individuals who are involved in the in signing the declaration, and we help them to link in to their ancestry, which they wouldn't have been able to do before. So being able to link into genealogy, into pedigrees when you don't have evidence is one of the reasons for testing.
Ali, we didn't see the whole screen there, your other screen, we just saw the PowerPoint.
Did you? Oh, the whole time there?
Yeah.
Oh, okay. Thanks for letting me know. Well, we can come back to this at the end. Can you see that now?
Yeah.
Yeah. That's the Declaration of Arbroath website.
Oh, no, we can't see that. No, we can just see the PowerPoint.
Oh, right. I don't know why that is. I'm sharing my screen. Right, well, we'll just go back to the PowerPoint anyway. There's something don't know why that is, because it should show. Let's be back to the PowerPoint now. Is that okay?
Yeah.
Yeah, we'll just go on from there. So DNA testing isn't a quick fix, folks. It's a process, and it's something that may well throw up new questions. It may well affect other family members. I've emphasised this earlier today in the other chat session we had, is that your DNA is not actually personal because you share DNA with close relatives. And that will affect how you reason when you think about how you share your own results and how you participate in. And I always like being upfront about that your DNA affects other people, because we've got to be aware of things to do with privacy and confidentiality. So many people have taken a test. I know from the questionnaire, there's a good number of you have taken the autosomal test with companies such as Ancestry, and somewhere in the region of 26 million samples have been taken around the world. The Y chromosome test, which is used for surnames, and I'll explain more about the difference in the tests as we go on. Only 750,000 tests have been taken. And the female equivalent of that is the mitochondrial test. Somewhere in the region of 200,000 full genome sequences have been done at Family Tree DNA. But the disappointing, I suppose, figure is at the bottom, and that's the MacLennan project, where we've got 95 people in the project. And of those, only around about 30 or so have taken a Y chromosome test. So of all the men called MacLennan in the world, only about 35 odd have taken a test, which compared to other clans such as the McRae's or McKinseys, we've got a lot of work to do to find out more about the clan. I'll come back to the clan later on, but for those who know nothing about testing, who are worried about giving a blood sample, there's no blood. It's a simple mouth swab that is scraped from the inside of your cheek, and then it's put in a wee vial and it's sent back to the lab. Other Ancestry use a spit, so if we actually find that some older people have difficulty producing spit, and then that's where the scraping actually works better. So the sample, as I said, is quite straightforward to take. I'm going to mention mitochondrial DNA at the beginning, and I won't really be talking about it again, just to make you aware of the different types of test. So the female test, we might call this, it's transferred down the female line from a mother to her children, and men inherit their mitochondrial DNA from their mother, but they can't pass it on. It's very much a female test. So it tracks back all the way into the past, down the female line only. And mitochondrial DNA is what powers our cell. Each cell in our body has hundreds of mitochondria in it, and each mitochondria has a full genome. It's different from the DNA, that your nuclear DNA, that your 23 pairs of chromosomes. It's completely different. And it actually came about in the bacterial soup that there was a cell, a single cell and organism, and a parasitic cell got into the cell in the primordial soup, you could call it, and survived. And that is how you have a whole genome of a completely different organism within another organism. And that's how we have mitochondrial DNA, and over many years, how they worked in synergy together. So anyway, that's a new fact I hadn't talked about this morning. So, so mitochondrial DNA is very small. The genome is only 16,500 bases, and that means that the opportunities for mutations is much smaller, because basically there's lots of genes in that genome, and if a mutation occurs within a gene, you'll have a genetic disorder. And so if it is a genetic disorder caused by a mutation in the mitochondrial DNA, the chances are the child or the fetus won't survive. And that's why it's quite limited in the number of mutations. They have to occur outside a gene for it to be transferred down the line. And because it's so small, the time frames for genealogy, they're too distant to be really particularly useful for genealogy. the mutations occur too distantly. But they have been used, and you'll know this example is Richard III, and he was found in a car park in Leicester, and the DNA from the bones, along with a number of other procedures from other disciplines, such as radiocarbon dating and analysing the bones and so forth, were used to confirm that the bones were those of Richard III. So the mitochondrial DNA that was found in the bones, Richard III here, you'll see his sister was Anne of York, so they shared the same mother. And the descendants of Anne of York, there's a number of maternal lines that survive to the present day and have very good genealogies, and they were used to corroborate that the mitochondrial found in the bones was the same as descendants of Anne of York. So that's an example where it can be used, but these are individuals who had a good pedigree, and most of us are female lines. They change surnames and then they die out, obviously, around about the 1780s when parish records cease, unless you're from other locations where the parish records are better, and then you link medieval genealogies and you could be very fortunate. But most of us, we don't have that. So mitochondrial DNA can show that we're connected to people, but it's much more difficult to establish when we were connected. There's certain instances where mitochondrial DNA can be useful, but we would need to speak about that on another occasion. So the other type of DNA that doesn't get broken up when it's passed on, so mitochondrial DNA is non-recombining, and so is the Y chromosome. That non-recombining simply means that it's placed, it's inherited intact as a complete unit, so to speak. So Y chromosome follows the direct male line, and males pass it on to their sons. So unfortunately, women, you don't have it. Your brother can pass it on to his sons. So if you want to research a surname, you need to get a male relative who would carry that Y chromosome, and we can use the Y chromosome for surname research because surnames, certainly in the Western world, are passed down the male line, and so they run in parallel with each other, which is a really powerful coincidence. A little bit about the MacLennan project itself. As I said, we're very small. We have 4 administrators. Bruce, you see there, Alice, who's been involved for a very long time, and also Anne Starbanger, who is the admin actually of the McKinsey project. And she's very knowledgeable about that family, and it's good to have her on board as well. The results are only there's mitochondrial results, but there's also the main results are the Y chromosome results. If you've taken an autosomal test, the results are not public. So we have about 32 men so far who've taken a surname test, Y chromosome test. We've got about 28 other men who carry other surnames who've joined for various reasons. Some who feel that they should be associated with the clan name. Others, they've got a female connection. What actually happens is that when somebody joins the project who might have a mitochondrial result, the Y chromosome is transferred over as well into the project. So that's why we often have, you see a lot of other surnames at the bottom of the listings. It's simply to do with the multiple types of test. And the project has an activity feed, but where you can comment and individuals can participate in discussion, but it's only open to those who are part of the project. So what else we can say about the testing a surname? Family Tree DNA is the main company that we use. Ancestry, who a lot of you will be aware of, who you use for your genealogy research, they don't do a Y chromosome test. They used to, but they stopped doing it. And so Family Tree DNA thus have 99% of all the surname projects. They also have the haplogroup and geographical projects. And these tend to be projects that look at beyond the paper trail. It's the common origins of lineages, medieval lineage research, so forth. The most important thing if you're taking a test is a relational database. So obviously if you've taken a test with Ancestry, you've got a massive database there. There's lots of other small companies about, but we're going to look at companies in a minute. It's best sticking with the major vendors. So this again just shows you the MacLennan project results for the surnames. I know it just looks like a whole load of numbers, but actually behind that is linkage and linkage of genealogy. So forget the numbers at this stage, just look at the different bands. So we've got about 16 different MacLennan lineages here. Because the MacLennans have had very little research done on them as far as testing, we don't have dominant lines appearing. This is very similar to most clans and how clans worked in the past. You have lots of individuals, men who live in a particular area. They come from different backgrounds. They take the name of their feudal superior or the local clan chief, and they are the clan. The Y chromosome, mind you, is only a small part of who you are. So although you might not share the same Y chromosome as another, say MacLennan, we're all interlinked through different lineages. This is just one part of the evidence, and it's important to keep that in mind. So just because you don't match somebody doesn't mean to say you're not part of the clan. Clans were a diverse organism, and there was multiple linkage, intermarriage in different lines. So you can see that we've got a lot of work to do to identify and understand what the lineages are of the MacLennans. But we're beginning to get there. There's a couple that are we've got 4 participants in each group, but there's a lot more to be done. So the main test that most of you who indicated on the questionnaire have done is the autosomal test. Now, autosomal DNA, that's your 22 pairs of chromosomes, not your 23rd pair, which is the sex chromosome, which is the Y chromosome, and the X. The X chromosome, I'm not going to cover today. The X chromosome, women, you have 2 X chromosomes. You get one from your mother, one from your father. Men, you've got one that you got from your mother only. And they have specific paths of inheritance that are very useful. But the autosomes are the chromosomes 1 to 22 are found within the cell nucleus. And you get so much of your DNA from each of your ancestors. So 50% from each parent, 25% from your grandparents. Actually, your grandparents can vary from 19% up to, say, 29%. It's not an exact 25%, and that's to do with the way that the DNA is recombined in the egg when it's fertilised and so forth. We won't go into all the ins and outs here. But you can see that the individual at the front has got sort of a multi-banded chromosome. It's just showing one chromosome here, an example, the different colours that have come from different great-grandparents. And basically you get a shuffled mixture of the DNA of some of your ancestors. And that's really important to understand, because when you come to matching, you'll be wondering, why do I not match such and such a cousin? Or why, if you're wanting to research a particular line, you have to identify the DNA that's come from a great-grandparent. And there's ways that you can do that we'll go into. So recombination is a term we use just to say that the DNA has been shuffled randomly and re-put back together. So the companies, now this is a question that has been asked, which is the best company to test with? None of these companies is bad. I'd say the one company that is disappointed in the last year has been Living DNA. Down at the bottom there, they are a British company. They promise much, but they've not delivered. But they do have one part of their what they offer, which is, I'd say, better than the others, and that's to do with ethnicity, ancestry, admixture, which we'll come to later. The biggest company, obviously, is the big box there, is Ancestry. Up the top there, we've got Family Tree DNA. MyHeritage offer a good range of tools, and then 23andMe 23andMe, you've got to be mindful, is most of the people who have tested there have tested for medical reasons. So they're very reluctant to get engaged with questions on genealogy. They just don't reply. So it's a big database. So it could be that only certain people have tested there, and it might be that you might want to think about that. But they do major on medical types of results. These testing companies, the tests are really quite cheap. So if you wait for a sale, you can get a test for $50. They vary in price, but they're round about that. But the overall price actually goes up because, as I'm going to explain, you need to test more than just yourself. There's a sale on, I'm just highlighting this, a sale on at Family Tree DNA at the moment. So if you Google Family Tree DNA, you'll see there's a sale on. So testing strategy. If you're wanting to save money and wanting to get your results into as many databases as possible, then this is a strategy that I would suggest. There was a question that was asked, can you transfer results? Well, two companies you can't transfer into. So Ancestry and 23andMe, you can't transfer results from other companies in there. So you've either got to start with one or the other. So if you start with Ancestry and did an autosomal test, you're in the big database. You can then get your raw data and transfer it to these other companies, Family Tree DNA, MyHeritage, and Living DNA. Some of them, there's a small fee. It's not very much, but the others, to be able to access all the features, you might have to pay a small fee again. But being in the Ancestry one, I'd say, is to be recommended simply because it's so big. It's like 16, 20 million people. And the others are much smaller. While FTDNA has the ability to have lots of different types of results. So you can have a Y chromosome, a mitochondrial, and your autosomal on the one place. And that's an advantage. Okay, we'll move on from time's flying. Some common questions I want to bring up. The accuracy, they're all accurate within the parameters of the type of test. They're accurate, but one of the major problems is like documents. Documents are accurate in themselves, in the sense you can see the facts, but are the facts correct? And can you misinterpret the facts? You get a death certificate, and if you were to just take the parents' names on the death certificate at face value, well, it might be right, but the informant might have given the wrong information. So you've got to be, likewise with DNA, the actual results are accurate, but how you interpret them, you could overinterpret a match thinking because it tells you you're a genetic distance of 2 or something like that, oh, that must mean it's a very close match. But if you've done a low resolution test on the Y chromosome, that's not accurate in the sense that it's only telling you you're actually related. So there's a degree of knowledge required of how to interpret results, and that's fine. Any thing we learn, like driving a car, you have to learn the basics and how to do certain things. So another question was asked was to do with ethnicity results. We'll talk about that later on. Is there such a thing as Scottish DNA? Well, yes and no. There's parts of the country that have DNA profiles that are specific to that area, but there's not really a thing called Scottish DNA as such, because as I showed earlier on, we're a mixture of DNA from all our different ancestors. So unless you lived within a very close community where there's a thing called endogamy. So endogamy means that cousins marry cousins, and then they marry cousins after generation. And so, for instance, the Jewish community would be endogamous. And that means when they take a DNA test, they will have lots and lots of matches and lots of close matches. But those close matches are actually much further away genealogically than the DNA would show because there's so much inbreeding has occurred. the same segments have been passed down the line. So for Scottish DNA, we're quite a mixture of people. We've got people that have come in over the last 1,000 years from all different ranges, and if they came in from, say, Flemish into Scotland, well, Flemish DNA is very similar to the DNA of Eastern Scotland for the last 4,000 years. There will be slight differences, so it's very difficult to break apart population genetics. Although, as we'll see later on, there's lots of advances happening, and we'll look at that. So, why should you take a DNA test? Now, genealogical problems obviously are when the documentary evidence is not about, where you have an illegitimacy, or you want to know where about in a particular location your line came from. A lot of people think, oh, that's what the ethnicity type test will tell you. Unfortunately, they only give you a very broad brush. You have to be much more specific and test particular types of DNA to know where your particular ancestors came from. So we have to remember to always do the genealogy. A lot of folk are thinking, I can just do a DNA test at Ancestry, it'll throw up all my genealogy, and that'll be it. I don't need to do any hard work. It doesn't work like that, I'm afraid. We still have to do traditional research, and the DNA will inform on deeper ancestry, which is great, which we could never know about before, but the hard work still needs to be done. So, the thing about DNA is that it's objective. A lot of our documentary evidence or lack of documentary evidence means that some people might take guesses or might think, oh, it's good enough. Well, we want to be a bit more than good enough. In genealogical research, we don't seek to fit the evidence into our theory. We seek to get rid of all the evidence that doesn't fit in, that can't stand up to scrutiny. So objective evidence is something we're looking for. DNA, because it's been passed down the generations, is objective in that sense. But facts can be misinterpreted, and DNA evidence can be misinterpreted too. So corroboration is somewhere that it comes up quite a lot. People want to confirm that they're definitely connected to a particular line, and that is something that our DNA can do. So, a bit more about the issues that we can resolve. Illegitimacy is high up there. It might not be you. It might be a parent or a grandparent or a great-grandparent. And with a research strategy, testing the right people, identifying the right people in the first instance, then these brick walls can be overcome. One of the things that happens a lot in the records is that you come across families where the spouse is changed, where there's been a death, and there's lots of young children, and you're never 100% sure, are the young children of the earlier spouse or of the second marriage. And that is one of the reasons that people don't match when they do DNA, is that they've got a surname and they don't match other people within the line, and they think there's been an illegitimacy. Well, sometimes there might be, but there could be a hidden adoption of young children, because it was very common for men or women to remarry soon after the death of a partner. It was economic survival, and a family unit was much more secure with two bread earners, or one bread earner and somebody looking after the kids. Historical questions can be answered if you're able to identify a genetic connection to a gateway ancestor into medieval genealogy. You can identify that you're linked to particular lineages, people who signed the Declaration of Arbroath, for instance. But like any line, or any research, we often want to know where we're from, and be able to, more than just say that, we want to actually prove it or have evidence of it. So where are my MacLennans from? What part are they from? Applecross? Are they from somewhere else? Whatever. Well, we can, if the documentary evidence is lacking, we can use the DNA to connect with others who know where they're from. And that's why it's so important to get Scots to test, or people who know where their ancestry goes back to. Another more recent, not problem, but scenario is those who are now adults who were created by donated sperm and eggs, and they're now using DNA to identify what their backgrounds are, and obviously adoptees as well. And there's no such thing as being anonymous now. So if somebody submitted sperm, donated sperm, they can't be anonymous, even though they might want to be. So each test, it's really important to grasp that each test works differently. And that means that the DNA is sequenced differently, the comparisons are done slightly differently. Generally, the tests are all looking for similarities or differences, or degrees of those, and some use both. We're not using ancient remains generally, though ancient DNA is now and will be even more important in the coming years. The significant amount of ancient DNA research going on, the archaeologists, there's a lot of arguments going on actually between the archaeological community and how to use DNA, because it can destroy samples, but also how it can challenge their precious beliefs and what they have been talking about for many years, and then DNA comes along and might suggest otherwise about the movement of people and cultures. But DNA is informing genetic genealogy. We are getting more and more samples from the Neolithic, and there's it's like we're working from both ends. We've got the genetic genealogists are working from this end into the medieval period, and we've got the archaeologists who are working from 2,000 years ago and more recent, and they're getting samples, and we're going to meet in the middle. So how does the testing work? So simple mouth swab, put in the post, it gets sequenced. The random bases are then put in an order, and then the results are calculated. A comparison is done with other people in the database, and your results are posted up on a website, or some companies, they mail them to you, whatever. But the numbers, don't get hung up on the numbers as such. Just see that that's the evidence. There's other ways to interpret the numbers. So, but each number represents something of value. So, what do they look like, the test results? And I always like to be upfront. Some people are very disappointed with the results. It's not what they expected. Again, they expect the whole genealogy to be presented to them online. They won't have to do anything. Or they might not match who they expect, or there's not much matching, or there's too much matching, and they're just overwhelmed with the number of matches. It works all ways. So I would encourage you to see this as a journey, and to be patient, but also there's a need to actually be, I didn't put this in here, to be proactive, and proactive in the sense of encouraging other people to test who you think you might be connected to, or you could be speculative. I recruited lots of people in the north of Scotland who carried the MacDonald name, who lived in the same area where my father was born, and you'll see later on my grandfather was illegitimate, and by doing that I was able to establish beyond doubt the family name and the genealogy. So this is one particular company. This is Family Tree DNA. This is my homepage, and the three main types of tests are there. So at the top you've got the Y DNA. The big Y is another type of Y chromosome test for the surname. Then in the middle, you've got the autosomal DNA, and then at the bottom, MIT DNA, mitochondrial DNA. And each one of these tabs, if I was to click on it, it reveals more sort of information. Some of the information is much more detailed than others, but so the companies provide with some of the helps, but they don't go overboard. Everybody's DNA is unique, you know that. They can't write everybody's DNA like a script for telling you all about your history. We have to do a wee bit of that ourselves. But the main data is behind these tabs. So I get a certificate to show this is my Y chromosome. That means nothing. It's just a load of numbers, isn't it? So we have to go beyond that. And this is where a relational database comes in. You want to be connected with others. The autosomal test that many of you have taken, Ancestry present the data differently. So this is family tree DNA, and my mother, sister, my father and cousins, and my mum's first cousin down at the bottom there, Mr. Watson. And I'm going to show his result comparison with me in a second. Something else I'll highlight there is that along the top of the results there, there's a column, there's headings, name, relationship range, shared CM, longest block. And just highlight the shared CM, there was a question about how do we measure relationships. What is a centimorgan? CM stands for centimorgan, and I'll explain that in a minute. I've got it on a slide. But you see the figures that are below that. You've got 3,384 I share with my mother. Another relationship with my mum's first cousin down at the bottom there, 450. And you'll see it's predicted as first cousin once removed. Well, actually not prediction, I've written that in there. But it's predicted first cousin to second cousin range. The results are predictions. Beyond, it's to do with how much DNA is expected to be shared at each relationship. And there's a number of relationships that will share the same amount of DNA. And that's why we have to have a strategy of how to work out what is the connection. So autosomal DNA, centimorgans, it's not a physical distance. It's indicating how many times a segment of DNA, the probability of a segment of DNA being recombined. Now, that'll not mean anything to you. Just see it as the larger the centimorgans, the closer the relationship. You think on those sort of terms, that sets you in the right direction. And that's my mum's first cousin and myself. You can see the DNA we share on the different chromosomes, the blue colour. And we share 450 centimorgans, which is actually right in the middle of what we would be expected to share. That relationship. Now that relationship, we can view it. This is from a tool called the Shared Centimorgan Project. So you can see me there, right in the centre, Alasdair, it says self. Now, my mum's cousin is the box below. Because it's a generational difference, it has to be a distance away. So it would be expected that we would share around about 433 centimorgans. I actually share slightly more, 450. But the way that DNA is recombined at each generation, there's a random selection of DNA inherited, and it could be as low as 102 up to 980. So that's quite a significant variation you can see there. It could, if I share 450 centimeters, if I didn't know the relationship with Mr. Watson, it could fall and see these other boxes that are coloured, highlighted. The relationship could fall into any of those as well. So if you don't know how you're connected genealogically, just because you share DNA, you can't just assume it's one relationship. You have to work out what is the right relationship. And this one, because I know about the documentary evidence, it all feels fine. But if you didn't know, you'd have to do triangulation. You'd have to test other people to corroborate what the match is. And I would highlight at the other side there half relationships. Half relationships, that means saying somebody in your ancestry is illegitimate generally, and so you only share half as much of DNA as you would expect, and that completely changes the evaluations and needs a lot more thought. I'm not going to go into more detail than that. I'm just wanting to highlight you the need for a strategy for evaluation of results. So the right test to take depends on what your question is. If it's you want to find the right father, then a Y chromosome test is a good thing to take. But if you know that the father, who the identity of your father might be, and you've got another relation of that person to take to test, then you would get an autosomal test done. So it all depends on the actual question and how long ago the event occurred. You have to identify what are the various scenarios, and then seek to disprove the scenarios. You never seek to prove a hypothesis, because if you do that, you'll get the wrong answer, because you'll be blind to all the other possibilities. And genealogical research is about identifying the scenarios, the hypotheses, and then finding evidence that either backs up the hypotheses, and it basically disproves the other ones. So the other hypotheses all have to be knocked down, and that's how we should do the same with genetic genealogy. So don't skimp on testing. If you can't just prove a relationship or confirm a relationship taking one test. You generally have to test other family members. So first of all, if you have an elderly family member still alive, you ought to test them, because they have more DNA of your recent ancestors than anybody else. So elderly cousins, siblings are important to test, because they have more DNA of the DNA that you don't have. The threshold confidence levels is something important. That's particularly important for Y chromosome testing. Taking the cheapest test or the lowest quality test is not a good way to go. You ought to try to get the best level of test that provides the most data. So you need to evaluate all this. So you can join projects, you can collaborate with others. The short courses, education is really important, or seek professional help. You join projects or there's groups on Facebook. You've got to learn strategies, how to methods to evaluate the data, and there's tools that can help you with that, which we'll see. So I'm going to pause for a second, let some questions, and then I'll continue with the next part, because time's vastly running out. Any questions?
I have one, Alastair. It's Malcolm MacLennan. You mentioned that you couldn't import into 23andMe and Ancestry. I had testing done with 23andMe. Could that be exported into Family Tree DNA?
It could, yes, yes. You can export from 23andMe and Ancestry, but you can't import that. So yeah, you'd be able to transfer your ancestry, your 23andMe result into Family Tree DNA for sure, yes.
So the goal, if I understand correctly, is that Family Tree DNA is the database being used for the Clan MacLennan DNA. So it would be in our best interest to get data into Family Tree DNA.
Yes, yeah, that's correct, Malcolm, yeah. That's the best. Okay.
And then I wasn't aware that there were all different tests within the DNA profile.
Yeah.
I submitted to 23andMe for medical purposes.
Yeah.
Is it reasonable to think that the right tests are in the 23andMe database and that would be valuable for the MacLennan DNA project?
the autosomal test that you did with 23andMe, basically it's very similar markers they've used, so it can be transferred over and they will give you matching with other people in the database for your autosomes. But the Y chromosome test, 23andMe have done we call it a backbone test. So basically they just test certain markers and it's not very detailed. So you'll get a result, but it'll be like 2,000 or 3,000 years old, the marker. It's not genealogically relevant. So if you transferred your file in, the autosomal, the matching, cousin matching we'll call it, will be there. But you then need to do a Y chromosome test with family tree DNA. What they do, they just send out a kit to you, a sample, a new scraping. So it's quite straightforward.
Okay. And is there a slice of family tree DNA that is the MacLennan DNA Project, or?
Yeah.
So do we have to specify where we want our DNA results to go in Family Tree Maker?
No. You can if you're ordering a new test, if you're transferring, then you just transfer, and then there's a tab. You can then, once you've got your account, you can just join the project through the tab, which I'll show you in a minute. I can do that. If you're completely new to testing, then you can buy the test. As I say, it's on sale at the moment. We can join through the MacLennan project, and it means the results will show up there right away. But it's easy to join projects. It's very straightforward once you've got an account. I can actually, I'll do that just now, so people might be interested.
Yeah.
I'm hoping that this will show up now. It'll take a second to show, hopefully. Now, is that showing the results page, Bruce?
The same thing's happening. Yeah, the edit view of the PowerPoint is showing, not the screen.
Right.
So maybe minimize that one.
Right, I've minimized the PowerPoint.
Yeah.
Oh, it says pause sharing. Don't know why that is.
Don't know about that.
Can
you minimize?
Right, I don't think it's going to do it because it says at the top your screen sharing is paused.
Oh. Well, we can still see your screen.
Right.
Try clicking the middle. There you go.
Is that now? Yeah. Okay. So this is my home page again at Family Tree DNA, and up at the top left hand side you'll see some tabs, and then it's a join project. So just join project. There's a list of all the projects, or some of the projects will come up. Basically what you do is just type in MacLennan. You can see that there. There's 97 members. We've gained 2 members since this morning.
Good.
Great. We've had success this morning. So you just click on that, and I'm already a member, but there'll be a join tab at the bottom. It's quite straightforward once you have an account. So you transfer your results in from Ancestry or 23andMe. The links for transferring are in this presentation somewhere. But anyway, we'll come back to that, and I can also send the links to anybody that's interested. So we should be back to the presentation.
Alastair, is that
Yes.
a good point to mention about the privacy and sharing so that you can access the MacLennan results?
Yeah, the MacLennan project results, the Y chromosome results are public in the sense that the string of numbers is public, but each person has a whole load of privacy tabs that they can restrict what they reveal. So the results tend to be most of the results are public, but there'll be some that are hidden because the person's not wanting them to be shown more widely. One of the things with genetic genealogy, as I mentioned earlier, is to do with our DNA reveals information about others. And also, you might not want everybody to have access to your results, even within a project. So you can restrict what information is available. Your name will be there, but your address is hidden. You'll have an email address. I often advise people to have an email, make a new email address up that is maybe with Gmail, that is specifically for your DNA kit. And that way you know where any contacts are coming from. As far as information that's been shared, it's like Facebook. You only share what you're willing people to know about yourself, and your most distant ancestor is okay. But there's no need for you to share any other information, and it's hidden anyway from other individuals. Privacy and confidentiality is something that the main companies take very seriously, and I'm going to mention GEDmatch soon, and that's somewhere I would say not to, no longer to upload your results to because of data breaches. But data breaches can occur anywhere. We know that banks and you name it have lost data, and so all companies are potentially vulnerable, but the main testing companies, as far as we can see, are doing what they're meant to do. As far as GDPR, many of you will be aware of the confidentiality and so forth about sharing of information. The main companies are doing the right thing there. Has that answered your question, Melanie?
Yes, I just thought that was important for people to understand that if they want you to help them with their research, that they have to click to let you in to a certain degree of
I'll explain that as well. Yeah. Yes. So project administrators on, say, the project here, the project access levels that we have are set by the participants. So there's a limited, basically it's like private, there's limited, then there's advanced. So most people have it set to the middle one, to limited. That means the project admins can look at the results and can help with analysis. If there's something, if somebody's really quite old or they are not really technically minded, then they would maybe set their project access to advanced. That allows the admin to change things if need be, to download results, for instance. But those things can be tweaked, and test takers have control over those.
Alasdair, in the
Sure.
MacLennan project,
Well, yeah. Can you say that again? It froze.
yeah, in the MacLennan project, when it states a distance of 1, is that fairly close?
Okay, so that's a good question. So a genetic distance of 1, this is Y chromosome testing. How many markers have been tested? It'd be 37 or 67 or 111.
I'm not sure of that.
Right. Well, I can explain it. So a genetic distance of 1, 37 markers, which is the sort of the lowest, to present day lowest level of test. It could be close, but then again, it could be more distant. And we would very much recommend a minimum of 67 markers really for comparison. Even a genetic distance of 1 at 67 is, I'd say, more likely to be a close connection.
Oh, sorry, it's 111.
Oh, right. A genetic distance of 111. If it's the same surname, then, and you're from the same area, I would say yes. But this is one of the things with that particular type of test. I've done a lot of work with the Stewarts, and we see a thing called convergence. So convergence is where these type of markers are used for Y chromosome. They go away from each other over time and then they come back in. And this back mutation and parallel mutations of markers makes individuals appear much closer than they actually are. So we have the High Stuarts and the Bonkle Stuarts share common ancestor 700 years ago. And 111 markers, they can be only 3 genetic distance from each other, which would appear to be really close, but they're not. So it needs individual evaluation by somebody who knows what they're looking at. But the likelihood is, I'd say, more that they are close connected, but I wouldn't want to say that without looking at particular markers that have mutated, or to look at them within a group. And it's much better to look at a scenario like that in a group of other test takers, because then I can identify markers that have mutated more recently. That's a good question. Is John in the project?
Yes.
Yeah. Well, you can always email me and I can have a specific look at it. My email address is at the end of this presentation. You can drop me a wee email and I'll look at it for you.
Very good. Thanks.
I have another question.
Sure.
If we're looking, you were mentioning the dearth of participants in the Clan MacLennan Surname Project. If we are wanting to support that, or any of us in the clan are trying to expand that test result, is there any advantage to having other members, male members in the immediate family test when you already know that they're all descended from the same ancestor? Or is it really about expanding to the clan members that are not yet tested and a part of that project?
Yeah. If you're talking, Melanie, about Y chromosome here.
Y chromosome, the MacLennan surname. Yes.
Yeah. At this stage, I would say broadening the net is a good thing. We really need to try to get a better understanding of the different MacLennan lines, and then once we've got that, we can fill in the gaps a bit more. And there's a whole lot of MacLennans out there that need to be encouraged to test. And I was saying to Bruce earlier that individuals can donate to the project. There's a donation button. So if you can't test because you're not a direct line MacLennan in the Y line, a small donation into the group fund can then be focused. Some focused testing can be carried out to encourage people to test. So it's something that to think about. But I'd say broadening the net is the priority at this stage.
Okay, that's good advice.
So I'll move on to cover a couple of slides. I'm aware our time is up, but we'll just go. People were asking why we don't match individuals on our autosomal DNA, and it's to do with the fact that there's lots of, we lose DNA at each generation. So although we've got 1 million ancestors there 20 generations ago, we don't have that many ancestors. The number of ancestors who contribute to our DNA is only about 1,400. It's really quite a small number of our ancestors contribute to our DNA, and that's because of population collapse and recombination. And we just can't have the DNA segments from all our ancestors because the genes that are contained within the segments can't get broken up, otherwise the human race would come to an end. So they have to get transmitted down intact, and basically you can't keep subdividing segments, wee bits from everybody, and so we lose the DNA. So our DNA, we get half from our parents, half from each parent, and the genomes from each parent has to be cut down. And that means we lose DNA from our grandparents as well. So this is why we need to test siblings and close cousins, because that also helps us when we come to sorting out our DNA, what DNA comes from our father and what comes from our mother. And you can see that the percentages of DNA that we share with certain relationships. First cousins, 12.5%. But when we get further down, say look at third cousins there, it's less than 1% of your DNA you share with a third cousin. It's very small. And that means the likelihood of you actually sharing DNA with a third cousin is much reduced. And you can see this in this table here. Once you get to fourth cousin level, half the probability of you sharing DNA of being detected is less than half. But your sibling might share DNA with a fourth cousin. And when you're trying to work out how you're related to people, you need to test other people. And in this table, it just shows you the different cousin relationships. And I said before, well, basically the importance is of understanding cousin relationships. Visually understanding them is worthwhile learning. Each relationship there, you can see first cousin, second cousin, third cousin. There's a number below it. That's to do with the amount of shared centimorgans. But there's a range below that. So if you look at first cousin, it ranges from 396 to 1397. Quite a variation in the amount of DNA. And if you were to put in a first cousin amount, there are 866 centimorgans. There's these other relationships that would have to be considered. But this is all sort of theory here. You need a strategy on how to go about evaluating your results. And we'll come to that after this wee bit on ethnicity, which was a question that was raised as, is it worth investing or believing the results? How accurate are they? And I would have to say that they can vary greatly. And I would not really encourage you to invest too much in ethnicity results. They are companies base their percentages on a range of data sets. So they've gone out into the field and collected data. They've also used the data that they're of those who've tested with them. They've used academic papers, and these are all combined to give them a data set. Now, these data sets are improving all the time, and that's why your results will change. But many populations are very similar. So south of Scotland and North England, the genetic data looks very similar, same as Norway and Sweden, Poland and Germany. And you will not be able to really pinpoint. They're not used to pinpoint ancestors. They're just to tell you roughly how much DNA can be attributed to certain areas. And you shouldn't see it any more than that. The reason I don't like the word ethnicity is to do with the fact that populations were not homogeneous. They were a mixture of different people groups. And the admixture is a better word. So there are studies that are being undertaken by academics, and this one is being quite important in that the geneticists wrote it and the historians hated it. And I think the historians hated it because they were involved in writing it. But there's been quite a lot of online criticism of this particular paper. Not that it's wrong, but just that some of the language that they used was not approved of by historians. But this uses a technique that is used by one particular company, Living DNA. And you can see the similarities and differences between different parts of the country. In particular, you can see, you look at the Orkney Isles there, each island in Orkney's got distinctly different DNA. And this matching, this shows historical populations. So in the northeast of Scotland, you see what ultimately would be the Picts, and Angus and Fife and Perthshire, you see clusters that look very similar. Those in the southwest of Scotland and Strathclyde and Lennox look very similar to those in Northern Ireland.
Ali, could you want to put your pointer on? Can you use
oh,
the
yeah.
laser pointer?
Yeah. Right, I don't know what I did there. Right, can you see my pointer?
Yeah, mouse, that's okay.
Yeah. Up in Lewis here, quite a distinct difference. It looks similar to some of the populations in Orkney. Skye, again, looks quite different from, you can go by the colours. And down in England, you can see the Anglo-Saxons in red, quite a distinct population. But these studies are gradually beginning to filter into our understanding of genealogy admixture. This is my results from 5 companies. They're all different. The best one is number 2, and the can you see that number 2 has got a lot more sort of subdivisions. That's Living DNA. Living DNA are very good at this, but the other aspects of their testing is that they don't have much functionality to do with cousin matching, and it's a much smaller database. But for biogeographical admixture breakdowns, they're significantly better than the other companies. So we have 23andMe. Number 3 is Ancestry. MyHeritage is number 4, and number 5 is Family Tree DNA. Family Tree DNA are going to be upgrading their breakdowns in the autumn, later on in the autumn. But yeah, you can see how they're all different. My Living DNA, there's some percentages there I just cannot not make head and the tail off. My biggest one is Northern Ireland and South West Scotland, and I've got nobody from Glasgow. I've got a couple of lines, distant lines from Ayrshire. So I don't know how they managed to get that, because my mother's sides are sort of Fife and the Lothians. So I don't put a lot of trust in that. But some of the other breakdowns look better. So we'll move on from there. I just wanted to highlight that to you.
It said, Ali, that there's a bit of a commonality across to the Iberian Peninsula. But, you know, looking at those pictures there, they don't seem to show that.
No, yeah. But Y chromosome results, we can pinpoint migrations much more accurately from genealogy results. The autosomal results, the ethnicity, are very ham-fisted, really, and I wouldn't at the moment put a lot into them. The academics who are using different types of sequencing and use things called scatter plot and you name it, and that academic paper, they got much better results, but that's not being used in genealogy tests generally. So so that these tests on ethnicity admixture are accurate, but they're only accurate to the data set, and they're not really particularly useful for most of us to do with genealogy. If you have a very mixed background, so you've got Italian or German and ancestors from other places rather than the British Isles, you will have better results or more interesting results. And if you've got, say, ancestors from Africa or China, then these populations will show up. But for most of us boring Scots, they're not particularly interesting at this stage. So we need to watch our time here, but why have I got so few matches? It could be you've not got relatives tested. It could be there's been an illegitimacy on your line. It could be you're not matching who you expect to match. So you've got to be considered that. That is one of the things with testing. It will reveal information that you might not be comfortable with, or some other family member might not be comfortable with. So you've got to be prepared for that. It can happen, but I've had people who test and they don't match their brother or their sister, or they find that granny played away from home. It happens. It's just one of these things we need to be aware of. It's human life, but as long as you're ready for that type of thing. But it happens when we do documentary research, isn't it? We find that there's an illegitimacy or something doesn't tie up. But these tests can help clarify, though, that what's going on. Yeah, obviously, you might not match people because the DNA is just simply not there because it's been washed out. What else? Your cousins and siblings, yes, definitely try to get other close family members to test because, as we'll see in a second, that allows us to phase our data. Now, when we're doing matching, we don't look at very small segments. Don't waste your time with segments that are 7 centimorgans or less. You need to be bigger than that. If it's X chromosome, because of the way the X chromosome is inherited, females it needs to be at least 20 centimorgans, and for men 10. You really want to start with your biggest matches and work out how you're connected. And that's 60 centimorgans and above. So the process that you have to go through is you have to map your chromosomes. It's quite logical in many ways. So we use a thing called DNA Painter. It's the easiest tool to use. DNA Painter basically means you get your data from your test, you basically download it, and then you copy it into this utility, and you can attach the matches to certain chromosomes. So you can then basically understand what DNA is from your father and what's from your mother. You then want to identify who else shares DNA on those segments, or who else within a group do you share common ancestry with. And these in common with matches can be identified by using various tools, clustering tools we call them. There might be a match that you have that is a close match, but you just don't know where they fit into your genealogy. Both my grandfather and my grandmother MacDonald were illegitimate. For a long time, we thought we knew who my granny's father was. In fact, my granny had 7 or 8 half-siblings or siblings that are in Canada, and I've now tested, or they've tested, the second cousins there, and we now know that they're not full second cousins. We're not full second cousins. So we now know that the father who we thought was the father of everybody isn't. So I still don't know who my granny's father was, but I've got various scenarios that I'm wanting to work out with matches, and we can use a tool called the WHATO tool. What it means, what are the odds? So what are the odds of us being connected? And that tool allows us to build a tree, put in the amount of shared DNA, and see if it actually is feasible. Now, these tools are all hosted on DNA Painter. So DNA Painter is run by a chap, Johnny Pearl, in London. He's a great guy, and it's a very, very small fee to use the tools. Some of the tools are free. So DNA Painter, chromosome mapping is what it's called. There's other tools available, the what are the odds tool and the clustering tools. These are what you have to use to begin to make sense of your matches. There's no point in just looking at the the tools the companies provide, because the tools that companies provide are not necessarily the best. So it takes time, but it can be done, or you can get somebody to help you to do it. I mentioned GEDmatch earlier on. Now, GEDmatch is where you can upload your data from any company, and there's lots of tools there that can help with analysis. Unfortunately, the GEDmatch has allowed itself to be bought up by a company that does forensic work for various law enforcement agencies. And then 2 weeks ago, they had a data breach. You could opt out of the forensic analysis comparison. And what happened was everybody was exposed. Everybody's results were open for anybody to see. And so I would no longer recommend GEDmatch because of that. And I think, unfortunately, they've lost the trust with a lot of people. But the companies themselves, there's one company, Family Tree DNA, they allow police to use their database, but you can opt out of it under your privacy controls. And so anybody actually who tests on Europe is automatically opted out because of GDPR. But the companies are all under pressure to be accessed for the data. And unfortunately, it's more a reflection of the poor processing that's been done in certain countries rather than these companies. The companies need to be a bit firmer, I think. But GEDmatch was a voluntary organisation set up, and things move on. So very quickly, I'm aware we've well over our time, but Y chromosomes we've talked about, and it's one of the main areas that we are involved with the MacLennans. Surnames were adopted for a range of reasons. Some of them are to do with being connected to a clan chief, but others it's simply to do with the individual lived on the lands of somebody who was either their feudal superior or was a local clan chief, and they took the name. And clans were not an entity where everybody necessarily was related to a clan chief. They might have been on female lines, but in the male line, there's a whole load of different lineages took on the name of the local chieftain, you could say. So you will get differences in matching. You will not necessarily match the same surname, and that's nothing to be worried about. That's just the way it is. There's two types of tests. The first test that Melanie brought up, and somebody else brought up, John brought up about a genetic distance of 1. The STR tests are what we used to use as an introduction. We still do, but they have limitations to do with back mutations and so forth. And we now use SNPs an awful lot more. And particularly at the bottom there, you see the big Y 700. So rather than testing 37 locations, the big Y looks at 15 million locations. And it's very powerful. SNPs are the way ahead. SNPs don't tend to back mutate ever, while STRs do change. And STRs, it's good that STRs do change because it allows us to work out genetic distance, but they can change too often. So there can be limitations there. So in traditional genealogy, we build trees, and in genetic genealogy, we do exactly the same. So there's a tree. Don't look at the numbers, just look at the structure of the tree. You can see branching, and you see surnames at the bottom. And so that's all we do. We use genetics to build trees, and then we can lay genealogy on top of that. So at the bottom left-hand corner, you can see the clan chief who tested at Bannockburn. We also have, a bit further along, the chief of the Forsyths. We also have the Armstrongs, a group of Armstrongs from the borders as well. And this shows the historic movement of this particular line from southern Scotland up to the Highlands over a period of time. just like the Mackenzies are from the southwest Scotland at one time. The genetics confirms that. So as the Gaels moved up from southern Scotland and spread all over complete of Scotland, so did the lineages. So I got involved with testing because of my father being illegitimate, not my father, my grandfather, and DNA has allowed me to confirm the surname and confirm who else I'm related to. So the short STRs, we call them, short tandem repeats. This is a test that most people would start with. It's quite straightforward in that we evaluate how many times a short sequence of DNA has been repeated. So at the top line there, on the middle line, you've got male 1, you can see that, and there's been 8 times a small motif has been repeated, and in the results it just becomes an 8. So each of these numbers on the top column, not a column, is line, it says 12, 25, 15, 10. These are all just how many times a small sequence has been repeated. But the other type of test, the SNP test, looks at how many, one location, and it looks how one base has changed. So males 2, 3, and 4 have an A, and male 1 has a T. And that he'll carry that male with a T, all his descendants will have a T. So by testing a number of individuals, we can see how closely they are related. So here's 2 particular lines. One of the lines is mine, and the other one's of my closest match, John MacDonald of Strathclyde. His ancestor went to Suriname to be an overseer in the plantations. For many years, I corresponded with him, and I encouraged him to test. This time, I didn't know who, with my MacDonald line, was a MacDonald line because of the illegitimacy. You can see the results along the bottom there, that basically they're pretty similar. There's only 2 differences. So it indicates that we're pretty closely related. That's the geography. That's where my family stayed at the bottom there. Where the circle is, a place called Brekake in Caithness. And then these locations are where I've tested other people, and Strathclyde is the one at the top there. So the more people you test by just recruiting, what I used to do is I used to go into Ancestry or World Connect. I would identify other people researching MacDonald's in the particular location. I'd write to them. I'd say, do you want to take a DNA test? They'd say yes, because I was paying. And then you build up a profile of individuals. So you can see at the bottom there, they've got 5 test takers, and 4 of them are very similar. But there's one at the very bottom has different signature. You can see all the yellow, and that indicates we're not connected. So we're looking at patterns really as well. So by strategic testing, you can start connecting lineages. And as I said, Big Y is the new test. It's a discovery test. So what it does is it sequences a very large portion of the Y chromosome, 15 million odd bases, and it'll identify what value you have for all the known locations, the known SNPs, but it'll also identify variants that you carry that when somebody else tests will become a new SNP. So that's me in the red box there. And as Bruce mentioned at the very beginning, when he first tested, the 2 of us shared various values on our STR test. But now you can see that Bruce shares a branch with some McPhersons. And these McPhersons are in Lochaber, in Bray Lochaber, and they are also up into Lochelagan area. So we need more people to match Bruce so we can try and understand more about his line. But this lineage I'm using my lineage just because we've done a little bit of work on it. But we can actually see more about the history of the line now. So here we are. This is a different presentation of the results. You can see Bruce there at the right-hand side, matching some McPhersons, some Badenoch, and actually I don't have Bray Lochaber there because they just tested recently. Then you've got my MacDonald's, and then you can see as we work towards the left, it becomes this very much a focus on the Inner Hebrides. There's a one line that went up to Sutherland, and then at the far side we've got the McNeils of Barra. So using Y chromosome, we can understand more about the line beyond the paper trail and looking at medieval genealogy. And the McNeils here, they fit into a manuscript called the 1467 manuscript, which is a Gaelic genealogy of the clans. Funnily enough, the McNeil genealogy is only just been discovered within that manuscript, because it used to be thought that the MacLennans were recorded, but they're not. It was actually the McNeils. And that the McNeil genealogy there goes back to Bute, the island of Bute, and a chap called Ferchar of Bute who is recorded, and they're believed to be Norse. So that's showing a little bit about the new testing that is going on, and to understand all this, education is required. So there's a book by a chap called Blaine Bettinger. It's very good. He's on his second edition. There's the ISOG Wiki, which is down at the bottom there. You can just Google ISOG Wiki, and it has lots of information on all types of testing. Facebook, there's lots of Facebook groups on how to analyse your results. And then at Strathclyde, we run 2 8-week courses starting October. And these, they're called 8 weeks, so there's 8 sets of materials are released. There's a chat session and there's some online forums and things. Again, we covered much more that can be done in 8 weeks, but there's still an awful lot to learn. So there are also a couple other books. The book Tracing Your Ancestors Using DNA is one that I've been involved in, and it's a bit more advanced than the Blaine's book. We also have, somebody was asking about, is anybody doing a PhDs on Y chromosome? And there's not really. The University of Leicester do some research. It's something I would love to do, but we need to find a supervisor. So genetic genealogy is taught obviously at Strathclyde in our postgraduate programme. There's quite a few courses available in the US and elsewhere. At the very bottom there, you'll see ISOG genetic courses. If you Google that, you'll come up with a number of other courses that are available around the world. Most of them are online. And is there anything else here? I'm not going to talk about ancient DNA because I think we're running out of time. But your DNA will reveal how much Neanderthal you've got if you do a particular company. And most of us do have it. So I've covered a lot. We've asked a few questions. Now time for more questions.